my name is Reichelt. I'm doing my PhD at the Technical University of Vienna. And the topic of my talk today is aspects and implications of physiological process control. Short agenda, I want to talk about the usual motivation of bioprocess development routines. I'm going to outline the state of the art, how it is done in some industrial projects, and then give arguments on the physiological control, why would it make sense and why you should look into that. Brings me to two topics: which would be the appropriate variable, physiological variable to control, and how do you actually conduct the necessary online biomass estimation. And I'm going to conclude with a short outlook on possible applications in the summary. So the motivation is pretty much the same all the time. You want to maximize your productivity, and in respect of the quality by design initiative, you probably want to have a constantly high product quality. Of course, it's of high interest that your process is robust and that your scale up through over the factors of, 4,000 to 8,000 is stable. And if your gained knowledge is transferable from process A to process B, you decrease your time to market, and that's also very beneficial. So to the technical control, the main driver usually is simplicity. So you have a batch phase, a fed batch phase with an exponential feed, and an induction phase. So your biomass grows over the time, and most of the processes feature a constant feeding rate throughout the induction phase. So the usual routine for a simple bioprocess development would be that you vary the growth rates in your pre-induction phase. You also vary the length of your pre-induction phase, so you yield different biomasses at this point, and then you also investigate the impact of different feeding levels in your induction phase. So every 3 factors, you get this DOE cube, and you start your experiment, which results in some data sets like these. Here, the increasing biomass depending on the feed level you have throughout your induction phase. And then if you have a look at the specific growth rate, it declines over time, which is also pretty trivial since your biomass grows. What is not so trivial and a total difference to the pre-induction time is that your biomass yield declines in your induction phase due to the metabolic load. So if you think about reducing the number of experiments, you can combine factors. You can move from a technical factor as the feed rate in grams per hour to something more physiologically, where you simply divide the feed rate by the cell dry weight, and you end up with a specific substrate uptake rate. But this is the index init here because you only calculate this at the point of induction. You have a volumetric flow rate, and you keep that again constant. But the benefit is that you reduce your DOE by 1 dimension, and you save experiments. How that also was published by Wechselberger et al. In 2012. So this is a magic eye image I really like very much. If you keep looking at it for some time, and sadly enough we don't have that time, you're going to see a shark here. And the metaphor is for the QS is actually declining over time in your induction phase. So although you think you keep everything constant and everything well controlled, your cells who are actually producing your product are not in a physiological constant status. So here your QS, your specific substrate uptake rate declines. You have an uncontrolled delta QS over time. So that means although we have used this little trick of combining the factors to a physiological factor, we came across the finding that we have this hidden factor behind. We have a delta QS over time. And now you have 2 options. If you at least calculate a physiological descriptor, so you don't calculate once at the point of your induction time and use that as a response factor for your DOE evaluation, but you at least calculate over your whole induction phase a physiological descriptor, you get a hold of the problem. But it would even be better if you simply control your QS over time on a constant level. So that brings us to the motivation, where a problem statement popped up that we have undefined physiological status of our culture, a hidden factor, since we're focusing on keeping the reactor in a constant state, like constant feed, constant volume, whatever. But we're not putting the cells into the center of focus, although they are actually doing the work. So physiological uncontrolled features volumetric constant feed, neglects physiology, and thereby the process knowledge is hardly transferable. If you control your physiology, on the other hand, and dynamically adapt your feeding rate, you get the chance to actively control your cell physiology in terms of specific substrate uptake rate or specific grow rate. That enhances your process knowledge, and that also fosters your scale up and transferability of your findings. But now we come to these two questions: which would be an appropriate variable to control, and how do you actually conduct the necessary biomass estimation? So in a microbial process, luckily enough, you don't have so many physiological rates. We have QS, specific substrate uptake rate, and downstream the growth, the productivity, and the energy throughput. But since QS is upstream of these other variables, it provides energy and growth independently, and you can even steer or predict the productivity by controlling and predicting QS. So I'm trying to argument here for QS, but since literature has a slightly different opinion on that matter, I have put the slide in here comparing the QS control compared to the mu control. So as I have outlined in the beginning of my talk, the mu is decreasing over time since you have this metabolic load onto the protein production. So protein production competes with growth, that's why your ypsilon-xs declines. So if you keep mu constant, like in this graph up here, due to the declining biomass yield, you must push more and more feed into your reactor to keep your, theoretically, your growth constant. And that simply boosts your QS. So that is not really technically feasible. My second argument is that since you, for the calculation of a specific growth rate, you divide Rx by your biomass you have in your reactor. So you need your biomass estimation twice. Well, the thing is, the biomass estimation usually features the highest error of over all estimations you have online. So the mu prediction or estimation is more error prone than the prediction of QS, since QS takes instead of the Rx, Rs, so the substrate uptake rate. It takes the biomass only once into account, and therefore it's less error prone. If you keep QS constant, again, the ypsilon-xs decrease leads to a decrease of mu over time, but your in terms of substrate uptake rate, you're constant, and the approach features lower dynamics, and therefore QS control appears more technically feasible. So instead of keeping a flow rate constant, we now change over to keeping the physiology of the cell constant. But that requires that you actually know the trajectory or the current concentration of biomass in your reactor. So you have to estimate how much biomass you have in your reactor, and by this equation you can then simply calculate how much feed the reactor needs to a given time point. So you have the output is a substrate feed rate, since you have to adapt your feed for the biomass, and the required inputs are the biomass and the reactor content monitoring. So coming to the first principle of biomass or first approach of biomass estimation, we used the ypsilon-xs trajectory. So we took a lot of historic data and plotted it against time. So this is the first plot here, where you see hardly a correlation, and you really cannot use it, although it's data-driven for anything. Since the experiments were very different, we wanted to use one trajectory for all the processes we wanted to conduct in our DOE. But if you plot it against the cumulative consumed substrate, the thing looks different. It's not the best correlation, I'm not saying that, but it is a correlation which actually helps you to predict how much biomass is produced from how much feed. So you can calculate beforehand the feed profile, and from this feed profile you can calculate how much biomass you will have to a given time point in your reactor. The second approach is a permittivity-based approach. So you need a permittivity probe, and that uses the permittivity or capacitance of a liquid. So that's the resistance of the cells to a spreading electric field. in order to correlate the signal with the biomass. Since the cells have a transmembrane potential as long as they are alive, that works really well. So one thing is, one drawback of this method is that the permittivity actually correlates to the size of the condenser, so of your cell. So it actually correlates to the biomass volume, and you have to assume that the biodensity keeps constant, and then you can set biovolume to biomass dry weight to a constant factor. So your input, the required input is the permittivity signal, of course, and the calibration, which you would do, for instance, in the fed batch phase. And then you get a signal over your induction phase, which correlates to your biomass. The third estimation approach, which is our favorite, is the approach via a balancing. You estimate your Rx, so your biomass formation, in time increments based on balances. So you use the C balance, what goes in, what goes out, and the difference is pretty much what is bound in your reactor. And then with the second balance, with the electron balance, you can actually reconcile your estimation and therefore increase the statistic significance. And then you simply integrate your Rx over your given time increments and cumulate these time increments to yield the total biomass at the end. And the inputs are pretty easy to calculate or to measure. You need the feed concentration and the density. You need the stoichiometry of your biomass and the biomass concentration at the starting point of your soft sensor. And then you need to measure only the off-gas concentration of CO2 and O2. And, of course, the flows of gas and feed into your reactor. This is a very nice graph since it comes from industry. You see that by the huge sampling intervals. So that was a shift and that was a shift. If you have a lot of master students or PhD students, you would have more samples. But the point is that we already transferred the soft sensor approach in an industrial environment, and they actually managed to estimate the biomass online. So this approach is definitely transferable. We also controlled QS with this approach, so on different levels, low, mid, and high. And you can already see that the assumption that you have no accumulation is a very important one, since if you reach the physiological maximums of a cell, you run into accumulation and then your balances don't close anymore. So as long as your balances close, the biomass estimation approach works fine. Otherwise, you have to add an additional balance in order to also account for accumulation. So summing up the three different methods, the Y/X trajectory has a very low cost of implementation. You need no additional equipment or software. The accuracy can be regarded as sufficient, but it has a very low robustness. If anything deviates from your schedule, you can recalculate, but it's not a closed loop control. The transferability, as long as you have historic data you can set up your model with, you're fine. The soft sensor also features low costs. The accuracy is regarded as good. It's only dependent on how well your C balance closes. The robustness is high since it estimates in 5-minute intervals and checks the consistency. And it's also regarded as highly transferable. The permittivity features a little higher cost since you have to buy a probe, and these probes are approximately 10,000 euros. And the accuracy is relatively good as long as you manage to get a hold of a good calibration. The robustness is mid since it's highly sensitive to electric fields. So in case you move the preamplifier closer to your steerer, your signals already change. And the calibrations are not transferable. So that's why the soft sensor is currently our favorite. So now to the applications. The transferability of the application of physiological bioprocess control with a soft sensor is really high. We have tested it with E. coli, with different E. coli strains, from organism to organism to Pastoris, and currently we're experimenting using it in cell culture. You can also use the soft sensor approach in order to estimate the accumulation if you add an additional balance, for instance, the N balance. What the major benefit is that you have controlled physiological state, and that you can also use this additional degree of freedom that you can set a dynamic in a controlled fashion. For instance, this was our hidden factor before, but now this factor wouldn't be uncontrolled anymore. You could set this delta here in a controlled manner, and for instance, if your physiological maximum is decreasing, you can set a ramp to really run across this border. So, summing up, with physiological process control, you keep your process in a defined state. You maintain your physiological status. The concept is transferable to an industrial environment, and you can use it for basically any physiological factor you want. It eases bioprocess development since it adds an additional degree of freedom. You get clearly defined factors and have a high dynamic range. And you can either exclude or use it to elucidate the effects of unknown variables. The control dynamics offer an approach to further increase your process beyond the current way of optimizing with constant feeding rates. And that I'm not only talking from an academic perspective. We did this in course of an industrial project. Sadly enough, I'm not allowed to disclose all the details, but I'm allowed to say so much that when we did the constant feeding rates, different levels, and we calculated this, the physiological descriptors, so the mean over the QS over the induction time in the same manner, we saw a significant increase even if we had the same physiological level of QS, simply by controlling the QS, so keeping the cell in a stable physiological state. So my point is, we should definitely move from technical control to physiological control, since the cells are also doing all the work. Thank you very much for your attention, and I'm happy to answer questions. Yes?
Yeah, because in your calculation of the yield from the substrate to biomass, you didn't consider the formation of the byproduct or product.
So we did that. We kept these processes under the physiological maximas. So you wouldn't accumulate substrate. As long as you don't exceed the critical QS or the critical substrate uptake rate, you will not, everything will be consumed totally. So a basic assumption is that the accumulation term throughout the induction phase is 0. But you can use this strategy. So permittivity wouldn't be affected at all. Permittivity would work. The soft sensor would need additional balance. Then you can also really calculate how much you currently accumulate. And the YXS trajectory probably wouldn't work when you accumulate significant amounts of substrate. You're right.
Yeah, if there are some product was formed.
There is product formed.
But I don't see you calculated the product formation.
The YXS yield, the trajectory doesn't respect the product formation. That's true. But if you put it into, we produced 1 gram per liter of product and 60 gram per liter of dry cell weight. So the area you make is relatively small. And also our product is intracellular. So if we dry, we also get the product with our dry cell weight.
This is the intracellular product, not the...
Yes. Both. So all products we have tested so far were intracellular.
This product was including in the biomass?
Yes. It's controlled on a constant level.
Constant level?
Yeah.
During process?
Pardon?
During
through the induction phase.
So just by changing the feeding rate of glucose?
Yes. So we measure the biomass and then we calculate how much feed do we need to apply to get the same QS again. And so the feed rate is calculated every 5 minutes, and it changes every 5 minutes.
So the feeding rate should be a dynamic process?
It's not, since it increases, like here, so your biomass increases and also your feed rate increases. So it wouldn't do this, since your biomass, you don't lose biomass usually, except if you sample, and that's reactor content monitoring. But sampling, if you take the full culture broth, you decrease the volume, but you don't decrease the concentration. And as long as you calculate the concentration, you don't run into trouble.